so I want to thank the organizers for the invitation, and obviously everybody who's watching this as well for your attention. So I want to take you a little bit back to 2022. So it wasn't that long ago. So this is 2022 was when the paper was published, but this was the data set that we had a couple of years earlier. So maybe let's say about 6 years ago. So what we tried to do was to go to the public databases, so the European Nucleotide Archive, and try to gather all of the metagenomes that were available back then, right? And the only criteria was technical criteria. We wanted a minimum number of reads, but otherwise we took everything that was available, and we were really, really happy that we had crossed these 10,000 metagenomes. We had 13,000, so this was a huge number back in the day. And you can see here in this global map that the North America is very, a lot of dots over North America, over Europe, the coastal regions of China. But as some of the previous speakers, in particular Alejandro, also mentioned, that there were huge parts of the world that were missing. And as well, we could see that there were all of these different habitats, but there was a total of 14, and half of the samples were from the human gut. So this was the state of the art about 2020, right? But this is what we had, and so we built this catalog by taking all of these genes from all of these metagenomes, and then because often we will be sampling the same organism in multiple locations, we collapse that redundancy in a hierarchical fashion. So both first starting with perfect matches, so exactly the same sequence, then this species level unigenes, and then these protein families. And so these are our data sets that we made available. And then so today, because of the context, I'll mostly be presenting these as resources, as things that we catalog and that we make available for the wider community. But in the manuscripts, you'll also find often a lot of these ecological analysis. And so, for example, or about the structure. And one of the findings that we saw is that when we looked at how this clustering took place, most of the sequence variation was actually coming from a very small fraction of all of the families. And then you had this very long tail of families, including families of which we saw one single, there were singletons, we saw one single sequence that doesn't cluster with anything else. And then at the other extreme, less than 1% of the families already are responsible for most of the sequence. And so this was 2022, and then a few years later, we kept cataloging, and this is what the map looked like. And so now you can see, first of all, a lot more dots, and so we start to see dots over South America, over Africa. It's still a huge concentration over the wealthier parts of the world, but it's getting less bad. Let's phrase it that way. And also, it's no longer. We have more and more habitats. So here, in order to be able to display them, I had to group them into these broader categories. But we have many, many different animals have had their microbiomes sequenced. So we have actually at this point in time we had a total of 75 different habitats, which has kept growing. So the nature of exponential growth is that the nature of exponential growth is that every 2 years we end up sequencing as much as we had sequenced up until that point. So a couple of years ago we had 60,000 metagenomes, and then in the publication in 24 we had 100,000, and now internally we are getting close to 200,000, and this is now the biggest source of data that is available globally in terms of sequence variation for all sorts of analysis. And so we have over 1 million mags that cluster into 100,000 species. And so this is all made available as part of a database called Spire. And there's a website, and we've also now made available a Python tool that uses an API, so you can download. And importantly, and this is actually something that represents a tremendous amount of effort, a lot of the metadata was humanly curated. So even just that map that I showed you, just making sure that each of those dots is is where it's supposed to be. There's thousands of man hours of curation to make sure that we don't have a sample that supposedly comes from the ocean with the GPS coordinates of central Paris, for example, which happens all the time because maybe people make mistakes. Right. And so now so these are these big resources. I'll talk a little bit about one of our particular points of interest in our lab, which is looking at the small proteins, which was something that, with the exception of Amibat's group, most of the community had ignored until fairly recently. And this is because a lot of these tools, a lot of the infrastructure that the bioinformatics community has been developed since the 1960s, it just completely stops working when the sequences get too short. They're hard to predict. We cannot even tell you whether an open reading frame really is protein coding just from the sequence. Everything is very difficult. But our intuition was that if we see a sequence being repeated, that's by itself probably already something happening there. So we took the same data set I showed you before, and we did the same exercise of cataloging all of the small open reading frames with the one difference that any sequence that was a singleton, we actually did not consider it. Because we think that there's too high of a risk that is an artifact. But we also make available a tool where if you have a metagenome or a genome, if you see a small open reading frame there, probably by itself it's very hard to know whether it's meaningful, but you can quickly check whether it also appears in other data sets. Because one of the things we see is that we see these sequences that are very widely conserved. So this is both conserved in evolutionary terms, so across different phyla, also conserved ecologically, so in different habitats, so that we find them around the world, and we have no clue what they're doing. So we have this, this first there, there's a sequence, there's an identifier 287924202, very widely conserved, so that probably something biological is happening for evolution to preserve this sequence, but our tools simply do not work. So we've tried to tackle this by focusing first on antimicrobial peptides as one subset of small proteins. So this being, sequences that kill bacteria or at least stop them from growing. And then we work with Cesar de la Fuente's group at UPenn. And instead of using sequence homology, we use machine learning. And then we processed our data and we built a set of also a catalog of candidate antimicrobial peptides, which we then tested in the lab. So this is something that has worked actually quite well. And currently we are trying to expand beyond the antimicrobial peptide space, where we think that machine learning is one solution, but another volley is to try to use the genomic context to assign functions. And so here's, for example, one example. Here's this 53 amino acid protein, fairly well conserved across these nitrospira mags that we got from long read sequencing. And we cannot really tell you from the sequence of the protein what it's doing or give you any predictions. But if we look at its context, we can see that it's always in these defense domains, this UVRDC. It always sits in the genome next to it. So we think that this can now start to give us hypotheses at a broader scale about what are these proteins doing, right? So just to summarize, we have these catalogs that we've been building, and I say we, it's actually this informal consortium, my group, Sebastian Schmidt's group, Per Bork's group, Jaime Huerta-Cepos in Madrid, and we've built these catalogs of genes, mags, small open reading frames, AMPs, and they share these common identifiers. A lot of the manual curation of the metadata was also a huge amount of effort. But so here today I focused on these as resources. Everything's available, but the manuscripts also often have more analysis that you should check out. So as I said, this was not done just by me. I've mentioned several of the people here in bold. I also want to thank the Australian Research Council and then NHMRC for funding my research.